So the first really serious evolutionary theme we're going to explore is a branch of evolutionary biology called systematics. Simply put, it is the study of biological diversity and of relationships. One who practices this study is called a systematist. And there's basically three facets to systematics. One would be phylogeny, and that's what we're going to focus most on. Phylogeny is the study of evolutionary history. So folks who do phylogenetic studies try to reconstruct the evolutionary history of a group of organisms. Evolutionary histories are presented visually in the form of branching diagrams. We call them phylogenetic trees or evolutionary trees. And this is a big one. It is based on the three domains of life. So each major branch here essentially represents kingdoms within the domains. So in addition to phylogeny, systematists practice taxonomy. And this is the act of describing diversity and creating classification schemes. And the third aspect is nomenclature. And that is how we craft names for species. And we'll explore that in a little bit also. So I want to focus initially here on phylogeny and on taxonomy. So again, we use branching diagrams to portray relationships and histories. This figure shows us how to interpret a branching diagram. So there are taxa represented here. Taxon A, B, C, D, E, F, and G. The word taxon means a group of organisms at any level in a classification scheme. These taxa are all derived from a common ancestor. So if we go back to the base of the tree right here, that represents the common ancestor of all those taxa. The first taxon to diverge from the rest is G, right? So we see their branch coming directly from the ancestral node. And so we call G the basal taxon. Any taxon A through F would be more closely related to each other than any would be to G. We have another interesting point right here. This node represents the recent common ancestor of D, E, and F. Now, we do not have sufficient information to resolve the relationships between D, E, and F. It is highly unlikely that they all simultaneously diverged from one ancestor. It is more likely that within that cluster of three, two of them are more closely related to each other. But we don't have enough data to portray that, so it appears as what we call a polytomy, an unresolved branch. The two taxa that are the most closely related are B and C. B and C share the most recent common ancestor, and these are what we would call sister taxa. All right, so how was that previous tree generated, right? What was the basis for creating those branching patterns? Well, there are different sort of schools of philosophy within systematics, and there are different algorithms used to take raw data and generate trees. And we're going to learn a little bit about a systematic approach called cladistics. The goal of cladistics is to recognize groups that are monophyletic, right? Mono means one, phyletic refers to evolutionary history. What we want to do is recognize groups that all share a recent common ancestor. So in cladistics, the monophyletic group includes the ancestor and all of its descendants, right? So that's kind of a unique feature of cladistics, is we include the ancestor, which is no longer alive, in the group. It's part of the monophyletic grouping. So here's a little sample tree, and we can draw a box around the branch that includes A, B, C, and their recent common ancestor, and that is a monophyletic group. It is not the only monophyletic group on this tree. In fact, a second monophyletic group could be created here by including D, E, F, and G plus their most recent common ancestor. And the largest monophyletic group is in fact the entire tree, right? So these monophyletic groups can be nested within each other. So those are really the only three valid groups we could recognize in cladistics. If you created a group that included, let's say, A, B, C, and D, but excluded E, F, and G, that is not monophyletic. That is a no-no. So sometimes when researchers conduct a cladistic analysis and they generate a tree and you compare it to pre-existing classifications, you might identify what was previously now recognized as a paraphyletic group. A paraphyletic group fails to include all of the descendants of a most recent common ancestor. So here's an example of a paraphyletic grouping. So again, let's say you had a pre-existing classification scheme that put D, E, and F in one group separate from G. But when you generate your tree based on your data in the cladistic mode, you realize that's not a valid grouping. That is a paraphyletic group because it must include taxon G to be monophyletic. We can also recognize groups that are polyphyletic. Polyphyletic groups, all the members do not share a most recent common ancestor. So again, imagine you had a group that was made up of A, B, C, and D. It was a family maybe, and E, F, and G was a different family, and you did a cladistic analysis and you generated this tree. You would say, that group 3, that's not valid. We need to rearrange the classification to reflect the evolutionary history and only recognize monophyletic groups. All right, so how does the cladistic approach work? What are the methods? Well, the first thing you have to do is to identify the group of organisms you want to study. We call that the in-group, and it can be as big or small as you want. But the fundamental question is, what is the pattern of relationship within this group? Cladistics is a comparative analysis. You answer the question of who's most closely related to who by comparing them to a closely related group called the outgroup. We use outgroup analysis. So basically, the two in-group taxa that are most closely related are the ones that are the most different from this outgroup. In lab number 6, we are going to conduct a cladistic study of members of a genus Quercus. Those are the oak trees. And our in-group here includes the white oak and the post oak and the blackjack oak and the red oak and the shingle oak and the pin oak. Right? So we have these oak species, and we want to know which one is most closely related to which other one. What's their pattern of relationship? To answer that question, we're going to compare the oaks to our outgroup, the American beech. Beeches are a sister group to oaks. That's a good outgroup. You want the outgroup to be as closely related as possible to the in-group. All right, next step is to select the specific characters you're going to analyze, or the characteristics you're going to compare. Often it's morphology. Morphology is a synonym for anatomy. Morph is form. So morphology is the study of form. Most of the characters we're going to use are anatomical. You can use molecular characters, DNA sequences, amino acid sequences, RNA sequences. You can use behaviors. You can use physiological characteristics. There are many different types of characters that can be utilized. So in our lab, and this is all, again, in our lab manual, we have a number of characteristics we're going to look at: leaf arrangement, petiole length, the leaf margin, the presence of bristle tips, fruit anatomy, the anatomy of the upper epidermis and the lower epidermis, seed germination traits. So those are the 8 characters or characteristics we're going to examine in our cladistic lab. So once we have identified the in-group and the out-group, and we have selected our characteristics, we set about the task of comparing the state of each character to the outgroup. We're in lab, take every oak tree and look at all those characteristics and ask, is it the same as it is in the beech, or is there something different about it? And we'll be coding the character states in a binary code. Give it a 0 if it's the same and a 1 if it is different. We call it the derived state. All right, so I want to explore this ancestral and derived character state bit a little more here. This tree is basically an a priori assumption we can make that all of the in-group are on one branch and the out-groups on the other, and both in-group and out-group share a common ancestor down here at the base of the tree. So if you have a characteristic that is the same in an in-group organism as it is in the out-group, the most logical explanation for that is that's what the trait was like in the ancestor, and they both inherited that ancestral condition. But if you find a characteristic that's different in the in-group than it is in the out-group, the assumption is a change has happened somewhere along the branch leading to the in-group. So we say that that change is a derivation. It is in the derived state. So prior to lab, I took the outgroup, the American beech, and looked at each of those characters and coded the character state. Whatever condition was present in the beach is the ancestral. Anything different from that is derived. So the derived states are coded 1, the ancestral states are coded with a 0. So we take every characteristic, we look at each in-group organism, we determine what the character state is, and then we organize all that data into a matrix. Here's an example matrix. In this one, the taxa are listed at the headings of the columns. This was a look at backboned animals, a lamprey, a bass, a frog, a turtle, and a leopard. The outgroup is a little marine creature called a lancelet. The characters they looked at was the presence of a vertebral column, hinged jaws, walking legs, and an embryonic layer called the amnion, and hair. So you can see any derived state is indicated in 1, any ancestral state is indicated in 0. Now, what is kind of the trickiest part of a cladistic analysis is taking that data matrix and using the pattern of zeros and ones to build a tree. When you build a branching diagram, a tree, you use the presence of shared characters in the derived state as the basis for including 2 taxa on the same branch. When you do that, you create a tree we call a cladogram. And there's a guiding rule in cladistics called parsimony, which means build the tree that requires the fewest events to have happened, the fewest evolutionary steps. Build the simplest tree you can. When you're done, what you have in your tree, in your cladogram, is a hypothesis about the relationship between those organisms. And hypotheses should be tested. The way you would test it is to generate a different data set from other characters, run through the whole process, make a tree, and compare the branching pattern, the topology of the 2 trees, to see if your hypothesis is supported or not. So this is figure 12 in chapter 20, where we take our data matrix and we use it to build a tree. And of course, they have arranged the taxa and the data in such a way that this tree basically builds itself. But anyway, what we do is look for the highest number of organisms that share a character in the derived state. So we see that the vertebral column character is shared by all the in-group. And that is the justification for creating the first branch here, where we split those animals off from the outgroup. And the unique feature of a cladogram is it actually places that character on the tree. It shows us the basis for creating this branch that separates these organisms from the other. Now, as we look at the data further, bass, frog, turtle, and leopard all have a hinged jaw, and the lamprey doesn't. So that is the justification for separating the lamprey from the other 4 taxa. And again, we make a second branching. We indicate on the tree the basis for creating this group here that we've separated from the lamprey. Walking legs are present in the frog, turtle, and leopard. So again, another branch is created, supported by that shared derived character. Only the turtle and leopard have an amnion, and hair is present only in the leopard, so that character would go on the branch leading to the leopard only. So that's a very easy example where, again, the tree is pretty simple to build. We may have a few glitches in lab where things aren't quite so straightforward, but that's part of the fun, is trying to figure out what is the simplest tree we can draw given the data we have collected. I included this figure because it shows how behaviors can be used in a cladistic analysis. So this is a cladogram of crocodilians, and it uses behavioral characters, the presence of shared behaviors. Something we must be vigilant to look out for is convergence. Convergence can complicate our attempts to reconstruct phylogeny if we confuse analogous characters for shared derived homologous ones. Here is on top a marsupial mole, and on bottom North American mole. So the two critters are very distantly related mammals, but they have both converged on the same lifestyle and have very similar adaptations, but not because of a common ancestor, because of convergence. So analogy and homology, those are important ideas in evolutionary biology. Structures are analogous if they are similar in function, but are of different evolutionary origins. Structures that are homologous have common evolutionary origins. And homologous characters might look very different as organisms diverge from a common ancestor and pursue different evolutionary trajectories. Homologous structures can wind up looking very different, or just as analogous structures can wind up looking very similar. Embryological studies are really vital in uncovering homologies. When you look at an embryo and you see the same structures being formed from the same clusters of embryonic cells, that's a pretty strong clue that those are homologous characters. In this embryonic series here, we have a reptile, a bird, and a human, and we see some homologous structures. All have a tail. Ours is quite reduced. We call it the coccyx. All have gill slits or pharyngeal pouches, although we don't have gills, right? But we all have these homologous features seen in the embryonic stage. Homology also exists at the gene level. So if you have the same gene in an ancestral species and that species diverges into two different species A and B, those genes are homologous. We would call them orthologous genes. Within a species, a gene duplication event can create a second copy of a gene that diverges. Within a species, homologous genes are called paralogous genes. So recall, one of the facets of systematics is classification and taxonomy. And traditionally, organisms have been classified in a series of increasingly broader sets. So using this leopard as an example, it belongs to the genus Panthera. It's in the cat family, and it's a carnivore, and it's a mammal like you. You have a common classification to this leopard from class onward. You are in the phylum Chordata. You are an animal. You are in the domain Eukarya. There are currently 3 domains of life recognized. That is the highest rank in the classification scheme. Here's another way of viewing it, using the gray squirrel as an example, right? So it's part of a series of nested sets that kind of spirals out from the species. Squirrels are in the rodent order. That's a pretty big order. And they're mammals like us, again, and share our classification all the way up to the domain level. Now, a few decades ago, the fields of phylogeny and classification merged, and we came to realize that the way we classify organisms really must be based on a phylogenetic tree. We started adopting tree-based classification schemes. So classification should reflect phylogeny. In many cases, the classification scheme proposed by a phylogenetic analysis will nicely complement a pre-existing classification scheme, but not always. Sometimes it gets messy and people do not want to give up older classification schemes, even when presented with very sound evidence that, based on phylogeny, that classification scheme just doesn't work. Yeah, so here's an example of how a tree meshes with the pre-existing classification system initially set up by Carolus Linnaeus, a Linnaean classification hierarchy. Here is an example of where a modern cladistic or phylogenetic based classification scheme clashes with a long-held traditional hierarchical classification. All right, so we in the past had a class called reptilia, and the reptiles would include lizards and snakes, crocodiles and dinosaurs. And then we had a class aves for birds. The class reptilia is paraphyletic. It fails to include all of the descendants of a common ancestor. Birds are clearly descended from Saurischian dinosaurs. So if a lizard is a reptile, so is a bird. So a more contemporary classification scheme would simply name each branch and not worry about where it falls in the traditional hierarchy. We call lizards and snakes squamates. We call the crocodile branch crocodilians. We call the remaining phylogenetic branch the archosaurs. And then, if you want, you can make a distinction between the avian and the non-avian archosaurs. So there is this adoption more recently of a rankless tree-based classification scheme where you generate the best phylogenetic tree you can and don't worry about which rank each branch represents. Just get the branching right and name each branch. So recall I mentioned that systematics has three major endeavors: phylogeny, taxonomy, and nomenclature. Let's talk now about nomenclature. In biology, we use a system called the binomial nomenclature. The images you see here represent three couplets. We have two organisms that at different parts of the world are both called corn. We have two organisms that in different parts of the world are both called bears. We have two organisms in different parts of the world that are both called robins. But clearly they're not the same species, right? Common names lead to confusion. So we have a system for giving every species a unique name. The history of this system goes back to the 1700s and a Swedish botanist named Carolus Linnaeus. We call it the Linnaean system. Um, Carolus Linnaeus was the name he used in his publications. His mommy called him Carl. His birth name is Carl von Linne. So typical of a scholar in his day. Latin was the language used to publish research, and he Latinized his name. The first time the Linnaean system was applied systematically was in a publication called Systema Naturae in 1758. This was essentially a catalog of all the known biodiversity at the time. And 1758 becomes a benchmark in biological nomenclature. No name using this system published before 1758 is valid. This is the date where it all starts for us. So, key to the Linnaean system is that each species is given a unique two-part name, or a unique combination of two names. There are very specific rules that a biologist must follow when crafting the name of an organism. In fact, in zoology, for example, there is a zoological code of nomenclature, and there's a botanical code of nomenclature. There is a congress that is assembled like once a decade to police the code, make sure the rules are clear, and arbitrate disputes about names. So one of the rules is that the name should be informative. The name should tell you something about the organism. This is the one rule that's probably followed the most loosely. People have fun with names sometimes. They might name a species after someone or in a whimsical manner, but the idea is the name should tell you something about the creature. Traditionally, the names were Latin, or at least Latinized. There's a lot of Greek used also, right? So Latin or Latinized. The first name in this two-part name is the genus. And the genus is a rank. It is a group of related species. A genus is a proper noun, so it should be capitalized. It is akin to your surname, right? Your last name identifies the group you belong to. And if you have siblings, you have other people with the last name, but you have a unique first name. Your first name would be the specific epithet. That's the other part of the name. The epithet, or the species name, identifies one particular member of a genus. So every species should have a unique combination of genus and specific epithet. Here's a couple of examples. We have the red oak, Quercus rubra, and in the same genus we have the willow oak, that is Quercus phellos. Here's another example of binomial, Campostoma anomalum. That is the name of a fish called the common stone roller. What we see here is a page from a book called The Fishes of Illinois. It's a catalog of all the fish in Illinois, plus a picture I fished off the internet, a colored photo. It's a pretty cool fish. First of all, this book, The Fishes of Illinois, is a pretty remarkable book. Look at the map of Illinois. Every dot there represents a site out in the field that the researchers went to and collected this fish, right? So in preparation for this book, Illinois was scoured from head to toe. Every creek, pond, lake, puddle was seined and sampled to get a comprehensive look at all the fishes of the state. So the common stone roller was originally named by a biologist named Constantine Rafinesque. So notice how after the name Campostoma anomalum is in parentheses the name Rafinesque. That's the author of the name. And the reason it's in parentheses is That is not exactly the name Rafinesque used. What we have over here is a list of synonyms that have been used for this fish. The very first one is Rutulus animalis. In 1820, Rafinesque described this new species. It was found in the Licking River of Kentucky. That's what we call the type locality. That's the original spot where the species was discovered. And he thought it was part of the genus Rutulus. The next entry there is by Nelson. In 1876, Nelson studied this fish and said, hey, you know what? This fish is really a member of the genus Campostoma. So he published an argument for why the name should be changed, and it was changed to Campostoma animalum. So we preserve the epithet. It had to be changed from US to UM ending to match the gender of the genus. So in biology, names are used by consensus. Enough people trusted or bought into Nelson's argument that this fish is really a member of the genus Campostoma, that's the name that started to be used, right? So it is done by consensus. In 1885, a fish called Oxygenium pulverentium was described, and as it turns out, was really a hybrid between a stone roller and another fish called a red-bellied dace. So that name can no longer be used for any species. So at the end now we have the final entry in that list of synonyms. The name we use today, Campostoma animalum rafinesque. Okay, a couple of follow-up notes about this fish and the name. So I told you the name should be informative. The genus Campostoma. Stoma means mouth. Campo refers to being curved, right? So the genus means curved mouth. So if you look at that guy, you can see he's got sort of a sad clown face. His lower lip is hard. They call them stone rollers because they flip rocks over and they scrape algae off with their lower lip. The epithet animalis, an anomaly is something that's unusual, right? So this fish has some unusual characteristics. So it means unusual fish with a curved mouth. Notice how I italicized the name. One of the rules is that in print, the name should stand out from the rest of the text. So we either italicize it or underline it. The common stone roller, that's its common name. Ichthyologists in the US have got together and decided on a standardized common name. Now they still use the binominal, but in communicating with the public and in less formal publications, the standardized common name is used. The bird people and the amphibian reptile people have done the same thing. This guy's a male. Those bumpilies all over him are called his nuptial tubercles. During the breeding season, he gets these keratinized spikes. They're kind of like antlers on a deer. They fight with them. It's a pretty weird looking creature. Now, since this presentation has dealt with kind of big level evolutionary and classification ideas, I wanted to take a minute and talk about biodiversity. The diversity of life on Earth is the product of almost 4 billion years of evolution. So we have an amazing amount of diversity. What we have today is just, the current version of evolving gene pools. In fact, most of the diversity of life on Earth is extinct now, right? So what we have is the current cohort. It's really hard to pin down how many species there are on Earth. I'm going to share with you a table here. It came from a United Nations initiative several years ago to assess biodiversity. And there is a column here for major taxonomic groups. It uses a kingdom as a rank. It was a tally at the time of how many species were described and an estimate of what we think is out there yet to be discovered. So check out these numbers for a minute, and you can see some pretty clear trends. I want you to appreciate the gap between what we currently know and what we think is out there. Pay attention to what's the biggest group, what's the smallest group, where are the gaps that are largest, where are the gaps that are smallest. So what we see here is the current head count is somewhere north of 1.7 million. I think if you looked around today, you'd see an estimate of close to 2 million species. That's how many have been described. This estimate says there is perhaps 14 million species on Earth. Holy cow! The number of described species is just a fraction of what is out there. Where are all these undiscovered, undescribed species, and what are they? Well, many of them are animals, probably not mammals, but probably insects. Animals are the biggest group known. So of that 1.7 million, 1.32 million of them are animals, mostly invertebrates, mostly insects. The biggest gap is in the bacteria, right? 4,000 described, perhaps there's a million out there because they're small and there aren't as many people studying bacterial diversity. The bacteria we do know about are mostly the ones that make us sick or that we use for medicine or industry. You could go out in your backyard and take a shovel full of dirt and spend some time, if you have a little microbiology knowledge, and you can find species that haven't been discovered yet. The smallest knowledge gap is in the plants, and they're macroscopic and they're pretty stationary, so that makes some sense. Now, so part of this gap is due to the amount of time and effort being spent studying the groups. So these come from some publications recently predicting richness and comparing it to what is out there yet to be studied. So let's see, within the bacteria, again, 99.1% of them have yet to be studied. Viruses, 99.5% of them have yet to be studied. Fungi, that's another big underrepresented group. 93.4% of fungi have yet to be discovered and described. Nobody loves the nematodes, poor nematodes. Roundworms, 95% of them have yet to be described. 80% of insects. This publication was, or this graph rather, came from a publication that combed through the biological literature and compared how many papers were published based on studies of certain taxonomic groups and compare that to known and expected biodiversity. So we can say, for example, that birds are overstudied, right? There's many more ornithologists studying and publishing about birds than there are entomologists studying and publishing about insects, right? So compared to how many insects there are, there are very few entomologists compared to how many birds there are. There's lots of ornithologists. So when people dream about being a biologist when they're a kid, they think, I want to study whales, or I want to study eagles. They don't think, I want to study gastropod mollusks, or I want to study book lice, or earwigs, or beetles, right? So, but we need people to study those, and for reasons we'll argue about, well, we have in the ecology unit, and we will again later. This lack of taxonomic expertise is a problem because we don't fully understand the ecological role of these understudied organisms. We don't know what potential they might have for us, how we might be able to utilize them, exploit them to be selfish, or simply understand why we should appreciate them. All right, that is the last slide in this presentation.